Hello, here's your lecture on the rest of the viruses. This is basically chapter 25, and we're going to start with dengue fever. Dengue fever is something that was only known in Africa for about 400 years and is now coming into the United States and many other countries. France reported its first dengue fever positive patient. It's not bad. The first time that you get it, 80% of people have nothing more than a mild cold or that silly flu-like illness. The problem is if you get bit by another strain of virus, if it's carried by mosquitoes. So if you get bit again and it's a different strain of virus, it can result in a hemorrhagic fever. And a hemorrhagic fever is when you just start bleeding from everywhere. So I found this nice little video. Let me show it to you. We'll see what dengue is all about.
For most of us, a trip to Southeast Asia usually means a relaxing holiday in an exotic location.
Unfortunately, some of us bring back a souvenir that can have fatal consequences, not only to ourselves, but to those around us.
Typically, about 10 years ago, we would have on average about 10 imported dengue cases. And now it's up to like 30, 40, 50 a year. Dengue has gotten worse and worse.
In 2009, I reported on an outbreak in Cairns.
In Cairns alone, they're spraying up to 300 houses a day.
One person died and thousands were infected.
It just overcame me all of a sudden. It was like being hit by a truck, and I really did feel as though I was going to die at one stage. The pain was just so much, and there was nothing I could do to abate that feeling of pain.
Unfortunately, there's not much in the way of drugs to treat dengue fever. Sufferers just have to let the infection take its course.
I was drinking lots of water and just lying there. There was really nothing I could do.
The Aedes aegypti mosquito is responsible for spreading the virus between humans. It draws up blood from an infected person and festers in the mozzie for 12 days. When it bites its victim, the mosquito transfers the virus through its saliva to the next victim. 400 years ago, dengue was only found in Africa, but now with global travel, we see it spread all over the world.
I think the most seriously affected country would be Brazil. They report hundreds of thousands of cases every year. Southeast Asia as well, throughout Indonesia, Vietnam, Thailand.
Australia's love affair with barley has meant the number of dengue cases brought back here from...
Here in the U.S., we've had cases in Florida, Texas, Louisiana. I'll show you a map in a few minutes. And of course, the biggest problem is it only takes one person to reach the country with the dengue virus in their blood, and for one of our mosquitoes to take that virus out of a person and spread it to someone else. This is why it's becoming a pandemic. So what are the symptoms? It's a sudden onset of fever, a headache that is behind your eyes, terrible muscle and joint pains, even though I can't spell. It's the other name for dengue fever is break bone fever. So that woman in the video who said that she felt like she'd been hit by a truck and the pain, it feels like all your bones are breaking. It has a skin rash that looks like measles, so they're very tiny spots all over the body. Only the difference is if you push on them, they don't turn white. They continue to be there because they're broken capillaries. Because this is a hemorrhagic fever, it damages the cells lining the capillaries. There are 5 different types, as I said before. You get infected with the first type, you get immunity to that type. You get infected with the second type, and what could happen is that your immune system goes nuts and you end up with a full-blown hemorrhagic fever. And many people die from this every year. So why can't I change my... Okay. So this is from the family Flaviviridae. It's considered an emerging disease in the U.S. You can see this. There were numbers between 50 and 528 million. That's a huge spread. It was first reported in the late 1700s, and we figured out it was a virus in the 20th century when we started realizing how small viruses were. And it's endemic, So endemic means it's naturally found in 110 countries. So you have to watch out where you travel. And this is just a great map that I found that says this is where they have found dengue fever patients, and blue is where they think it's going to spread. Now look how far north these mosquitoes have been. We've got a little bit in western Connecticut, Massachusetts, Vermont. That's just crazy. This is a tropical disease, but this is what's happening. Okay, this is a virus that is becoming a huge problem, also becoming a pandemic, also an emerging disease, and it's not in your book. This is not in your book, so this is the only information you're pretty much going to have, but I wanted to include it because people in the Caribbean are living with this, and it is becoming a huge pandemic. So in 2013, chikungunya virus was found for the first time in the Caribbean. It has spread like wildflower. So between late 2013 and 2014, over 776,000 cases have been recorded in the Caribbean islands. They're not that big. 152 deaths. There's been more since then. Again, this isn't transmitted by mosquitoes. They're not really picky about what kind of viruses they carry. It's fever and joint pain. While dengue fever can take 14 days, plenty of time for people to get on an airplane. This one's a little bit shorter, probably about 7 to 10 days. But it's the same thing. It's headache, muscle pain, joint swelling, and a rash. So you really need to get tested to find out what you have. And I just thought it was interesting that the name, chikungunya, means to become contorted. That's wonderful. So what would be a difference between dengue and this? You would say, do you feel like your bones are breaking? And someone would go, ah! So then you would know it was dengue as opposed to contorted with joint pain. All right. So the very first epidemic of the new century was SARS. It became pandemic within 3 months. It was the first pandemic disease aside from AIDS that the world had to deal with in a really long time. And of course, it took AIDS a while to spread, and SARS spread within 3 months to many countries. This was pre-Ebola, remember? So SARS is considered an emerging disease. But it's a coronavirus. And coronaviruses have been known for years just to cause colds. It's the second most common cause of a common cold. It's transmitted by respiratory droplets because it is a respiratory disease. There's no vector like a mosquito or a bat or anything. People are just the vector. But what happened was it originated in China, and no one knew where it was coming from. The Chinese authorities were keeping pretty mum about the fact that they had an epidemic on their hands, until someone, epidemiologists started saying, well, this is coming out of China. What's going on there? And they finally had to come clean and say, actually it was one scientist who came forward speaking in fear of his life, who said, yeah, we've had this for about a month and people are dropping like flies and we don't know what it is. So then what ensued is an unprecedented international cooperation to figure out what this virus was, where it came from, sequence it, and found out that it was a new mutation of a coronavirus that was capable of killing people. If you take a look at this graphic, I put it up here because the information's not that good. So I thought it might be a good exercise if you print this out and you read it and you try to make it better, because there's a lot of information missing. So you can take your book and you can see where some graphic artists really got things wrong. There's one that's even funny in here. If you tell me the most glaring mistake in this graphic, I will give you a point, all right, on your exam. Okay, the next respiratory disease is RSV, respiratory syncytial virus. This is a serious disease in infants. It's a lower respiratory tract infection, so it's just the lungs. The upper respiratory tract is considered to be your throat and your nasal cavities. This is primarily in children under 5. Syncytia means that the virus-infected cells are able to merge with healthy cells, create these giant conglomerations of cells. See this over here? And that's how the virus evades your immune system. It is the leading cause of fatal respiratory disease in infants worldwide, and it happens here all the time. Infection is postulated as a cause of asthma in the first few years of life, severe asthma. Vaccines just make it worse because you've introduced more viral material and the immune system goes overboard. Up to 98% of children are infected by the age of 3 in daycare centers, 125,000 hospitalization, 2,000 die. It's extremely contagious. And in adults, it's nothing. It's just a cold. And I'll tell you that when my daughter was about probably 3 months old, she started to have trouble breathing, and it just got worse and worse and worse over only a couple of hours, from the middle of the afternoon to about 7 o'clock at night, and congested until it was so bad that we rushed her to the hospital wondering what was wrong. And they admitted her. They thought, well, maybe it's early onset of asthma. It wasn't until they couldn't get her to breathe, like 2 hours later, she'd been on lots of inhalers and she was in this metal crib, it was horrible, that they did a nasal swab, which is the only way to, you have to do a serology test to find out that it's an RSV virus, and came back and said, yeah, it's RSV, and yeah, this could be bad. She could die. So why don't you just stay up all night and make sure she gets these albuterol treatments? So she was better in a couple weeks, but it's very frightening. And I've known many babies since then to come down with this disease, and it's important that you realize this happens to young children and to get them supportive care as soon as possible. If it doesn't resolve fairly quickly, they do have antivirals that they will give. Okay. HIV. By now, you know what HIV is. You know some of the opportunistic infections associated with it. We talked about how HIV infects the cell preliminarily. But what I want you to do now with HIV, your book has tons of information on HIV, but what I'll ask you to understand is, what is a retrovirus? What is the genetic material that it has? What does it do in our cells? So you may have to go back to your genetics, but basically it uses some enzymes to put itself into our DNA, into human DNA. What is the origin of the HIV virus? I've given you the page, so I'm asking you to do a little work here. I've given you some hints. Where was the origin of HIV? I think you're going to find it a little interesting, and I'm going to show you a movie in a minute to help you out with that. Consider why, since 1981, we don't have a vaccine yet. So in your reading, you should find out that HIV replicates terribly. It's like a broken copy machine. It does not make very good copies of itself. So we can't have a vaccine against something that's constantly changing. I want you to know what kind of a cell HIV specifically attacks. What are the opportunistic diseases associated with HIV? That's in your book. With HIV, associated with HIV as a result is tropism. So that's page 733. I want you to know the current epidemiology with some statistics. I want you to really know the problem with AIDS, because we don't see it in this country, and this is a genetic bottleneck for the human race. The human race is being infected. This is a pandemic. It's slow, so it's not in our sights all the time, but I want you to come out of this class knowing how much of a problem HIV is worldwide. So take a look at page 734. And then I want your opinion on Truvada. Truvada is not mentioned in your book because it was only just approved as a preventative of HIV. And I'd like you to just take a few minutes and Google Truvada and be able to put together a nice essay piece on the societal aspect of HIV, with a little bit of the biology of HIV. So let's take a look at this. This is one of my favorite videos. He is a virus hunter, Nathan Wolfe.
When most people think about the beginnings of AIDS, they're going to think back to the 1980s. And certainly this was the decade in which we discovered AIDS and the virus that causes it, HIV. But in fact, this virus crossed over into humans many decades before, from chimpanzees, where the virus originated, into humans who hunt these apes. This photo was taken before the Great Depression in Brazzaville, Congo. At this time, there were thousands of individuals, we think, that were infected with HIV. So I have a couple of really important questions for you. If this virus was in thousands of individuals at this point, Why was it the case that it took us until 1984 to be able to discover this virus? Okay, now more importantly, had we been there in the 40s and 50s, 60s, had we seen this disease, had we understood exactly what was going on with it, how might that have changed and completely transformed the nature of the way that this pandemic moved? In fact, this is not unique to HIV. The vast majority of viruses come from animals, and you can kind of think of this as a pyramid of this bubbling up of viruses from animals into human populations. But only at the very top of this pyramid do these things become completely human. Nevertheless, we spend the vast majority of our energy focused on this level of the pyramid, trying to tackle things that are already completely adapted to human beings, that are going to be very, very difficult to address, as we've seen in the case of HIV. So during the last 15 years, I've been working to actually study the earlier interface here, what I've labeled viral chatter, which was a term coined by my mentor, Don Burke. This is the idea that we can study the sort of pinging of these viruses into human populations, the movement of these agents over into humans, and by capturing this moment, we might be able to move to a situation where we can catch them early. Okay, so this is a picture, and I'm going to show you some pictures now from the field. This is a picture of a Central African hunter. It's actually a fairly common picture. One of the things I want you to note from it is blood, that you see a tremendous amount of blood contact. This was absolutely key for us. This is a very intimate form of connection. So if we're going to study viral chatter, we need to get to these populations who have intensive contact with wild animals. And so we've been studying people like this individual. We collect blood from them, other specimens. We look at the diseases which are in the animals as well as the humans. And ideally, this is going to allow us to catch these things early on as they're moving over into human populations. And the basic objective of this work is not to just go out once and look at these individuals, but to establish thousands of individuals in these populations that we would monitor continuously on a regular basis. When they were sick, we would collect specimens from them. We would actually enlist them, which we've done now, to collect specimens from animals. We give them these little pieces of filter paper. When they sample from animals, they collect the blood on the filter paper, and this allows us to identify yet unknown viruses from exactly the right animals, the ones that are actually being hunted.
Deep in a remote region of Cameroon, two hunters stalk their prey. Their names are Patrice and Petit. They're searching for bushmeat, forest animals they can kill to feed their families.
Patrice and Petit set out most days to go out hunting in the forest around their homes. They have a series of traps, of snares that they've set up. They'll catch wild pigs, snakes, monkeys, rodents, anything they can, really.
Patrice and Petit have been out for hours but found nothing. The animals are simply gone. We stop for a drink of water. Then there's a rustle in the brush. A group of hunters approach. Their packs loaded with wild game.
There's at least 3 viruses that you know about which are in this particular monkey.
This species, yeah. And there's many, many more pathogens that are present in these animals. These individuals are at specific risk, particularly if there's blood contact. They're at risk for transmission and possibly infection with novel viruses.
As the hunters display their kills, something surprising happens. They show us filter paper they've used to collect the animal's blood. The blood will be tested for zoonotic viruses, part of a program Dr. Wolf has spent years setting up.
So this is from this animal right here, greater spot-nosed guanin. Every person who has one of those filter papers has at least at a minimum been through our basic health education about the risks associated with these activities, which presumably from our perspective gives them the ability to decrease their own risk and then obviously the risk to their families, the village, the country, and the world. Okay, before I continue, I think it's important to take just a moment to talk about bushmeat. Bushmeat is the hunting of wild game, okay, and you can consider all sorts of different bushmeat. I'm going to be talking about this. When your children and grandchildren sort of pose questions to you about this period of time, one of the things they're going to ask you is how it was that we allowed some of our closest living relatives, some of the most valuable endangered species on our planet, to go extinct because we weren't able to address some of the issues of poverty in these parts of the world. But in fact, that's not the only question they're going to ask you about this. They're also going to ask you the question that when we knew that this was the way that HIV entered into the human population, and that other diseases had the potential to enter like this, why did we let these behaviors continue? Why did we not find some other solution to this? They're going to say, in regions of profound instability throughout the world, where you have intense poverty, where populations are growing, and you don't have sustainable resources like this is going to lead to food insecurity. But they're also going to ask you probably a different question. It's one that I think we all need to ask ourselves, which is why we thought the responsibility rested with this individual here. Now, this is the individual you can see just right up over his right shoulder. This is the individual that hunted the monkey from the last picture that I showed you. Okay, take a look at his shirt. Take a look at his face. Bushmeat is one of the central crises which is occurring in our population right now, in humanity, on this planet. But it can't be the fault of somebody like this. Okay, and solving it cannot be his responsibility alone. There's no easy solutions, but what I'm saying to you is that we neglect this problem at our own peril. So, in 1998, along with my mentors Don Burke and Colonel Mpudi Ngole, we went to actually start this work in Central Africa to work with hunters in this part of the world. And my job at that time, I was a postdoctoral fellow, and I was really tasked with setting this up. So I said to myself, okay, great, we're going to collect all kinds of specimens, we're going to go to all these different locations, it's going to be wonderful. I looked at the map, I picked out 17 sites, I figured no problem. Needless to say, I was drastically wrong. This is challenging work to do. Fortunately, I had and continue to have an absolutely wonderful team of colleagues and collaborators in my own team, and that's the only way that this work can really occur. We have a whole range of challenges about this work. One of them is just obtaining trust from individuals that we work with in the field. The person you see on the right-hand side is Paul DeLong Manutu. He's one of the best communicators that I've really ever dealt with. When I arrived, I didn't speak a word of French, and I still seem to understand what it was he was saying. Paul worked for years on the Cameroonian National Radio and Television, and he spoke about health issues. He was a health correspondent, so we figured we'd hire this person. When we got there, he could be a great communicator. When we would get to these rural villages, though, what we found out is that no one had television, so they wouldn't recognize his face. But when he began to speak, they would actually recognize his voice from the radio. And this was somebody who had incredible potential to spread aspects of our message, whether it be with regards to wildlife conservation or health prevention. Often we run into obstacles. This is us coming back from one of these very rural sites with specimens from 200 individuals that we needed to get back to the lab within 48 hours. I like to show this shot. This is Ubald Tamofe, who's the lead investigator in our Cameroon site. Ubald laughs at me when I show this photo because, of course, you can't see his face. But the reason I like to show the shot is because you can see that he's about to solve this problem, which he did. Just a few quick before and after shots. This was our laboratory before. This is what it looks like now. Early on, in order to ship our specimens, we had to have dry ice. To get dry ice, we had to go to the breweries, beg, borrow steel to get these folks to give it to us. Now we have our own liquid nitrogen. I like to call our laboratory the coldest place in Central Africa. It might be. And here's a shot of me. This is the before shot of me. No comment. So what happened? So during the 10 years that we've been doing this work, we actually surprised ourselves. We made a number of discoveries. And what we found is that if you look in the right place, you can actually monitor the flow of these viruses into human populations. That gave us a tremendous amount of hope. What we found is a whole range of new viruses in these individuals, including new viruses in the same group as HIV, so brand new retroviruses. And let's face it, any new retrovirus in the human population, it's something we should be aware of, it's something we should be following, it's not something that we should be surprised by. And needless to say, in the past, these viruses entering into these rural communities might very well have gone extinct. That's no longer the case. Logging roads provide access to urban areas. And, critically, what happens in Central Africa doesn't stay in Central Africa. So once we discovered that it was really possible that we could actually do this monitoring, we decided to move this from research to really attempt to phase up to a global monitoring effort. And through generous support and partnership scientifically with Google.org and the Skoll Foundation, we were able to start the Global Viral Forecasting Initiative and begin work in 4 different sites in Africa and Asia. Needless to say, different populations from different parts of the world have different sorts of contact. So it's not just hunters in Central Africa. It's also working in live animal markets, these wet markets, which is exactly the place where SARS emerged in Asia. But really, this is just the beginning from our perspective. Our objective right now, in addition to deploying to these sites and getting everything moving, is to identify new partners, because we feel like this effort needs to be extended to probably 20 or more sites throughout the world, to viral hotspots, because really the idea here is to cast an incredibly wide net so that we can catch these things ideally before they make it to blood banks, sexual networks, airplanes, and that's really our objective. There was a time not very long ago when the discovery of unknown organisms was something that held incredible awe for us. It had potential to really change the way that we saw ourselves and thought about ourselves. Many people, I think, on our planet right now despair, and they think we've reached a point where we've discovered most of the things. I'm going to tell you right now, please don't despair. If an intelligent extraterrestrial was tasked with writing the Encyclopedia of Life on our planet, 27 out of 30 of these volumes would be devoted to bacteria and virus, with just a few of the volumes left for plants, fungus, and animals, humans being a footnote. Interesting footnote, but a footnote nonetheless. This is honestly the most exciting period ever for the study of unknown life forms on our planet. The dominant things that exist here, we know almost nothing about. And yet finally we have the tools which will allow us to actually explore that world and understand them. Thank you very much.
Why did I show you that really long video with the strange virus hunter guy? Because there's a number of excellent points in there. One is that we knew HIV emerged from the jungle in the 1930s. It emerged because of bushmeat, which remains a problem today in 2014. People have food scarcity and they have to turn to animals that carry viruses that have never been exposed to humans before, and they find us really yummy. And what happens is first they stick a little bit so the virus is only transmissible between the animal and the person, and then once it gets really used to us through mutations and accommodation, then it starts to be transmitted from person to person. And it took millions of years for SIV, simian influenza virus, immunodeficiency virus, to become HIV, millions of years. And it could take 1 million years for it to adapt to humans, where it doesn't make us sick anymore either. In simians, it's not necessarily fatal anymore. But in the meantime, a lot of people are going to die, which is why I want you to be aware of the state of the epidemiology right now. The next thing I found interesting about that video is that they are monitoring for diseases that can emerge from the jungle. This is very important work. So if I were to ask you, how are we working to prevent further retroviruses entering human populations, that would be one of them. He talks about wet markets and SARS. A wet market is a market mostly in East Asia where they have animals and you go pick out your animal and they slaughter it for you for dinner. Lots of human contact with blood and animal products there for viruses to jump between species. And I also think it's interesting that it's nothing, we haven't discovered everything that needs to be discovered. There's tons of things out there. Go become a virologist. Okay, here's a nice little diagram of HIV. It has, you should be able to give me a nice lowdown of exactly how it gets into a human being, what happens with its DNA, how it gets back out of a human being, and mention the specific cell it infects. Here is how complicated it is. These are spikes that attach. It's got its retroviral RNA inside of there. I just like that. Okay, what's next? Measles. How do we go from HIV to measles? Measles is no big deal. We all get vaccinated, right? No, we're not all getting vaccinated. This is a re-emerging disease, and it's killing people, particularly children. So, check out the learning outcomes on page 737. That's what I want you to know. It's re-emerging due to vaccination. It's highly contagious by droplets. 98% of the population must be immunized to prevent transmission. The vaccine is 95% effective, has very few to negligible side effects compared to other vaccines. The complications are on page 737. They're not pretty. People do die from this disease. Children die from this disease. Look up Koplik spots. When there was an outbreak of measles in Ohio, it went undetected for a number of weeks because doctors are no longer trained to recognize diseases that were once very common. Once we've been vaccinating against a disease, we just don't train about it anymore because it's been overcome. So children were coming with these small spots around their mouths that look like these little tiny grains of red salt, and doctors didn't know what it was. They didn't know. So it took a couple weeks for the measles epidemic to be spotted, and then by then it's just hugely contagious and it was a terrible outbreak. So here's a little blurb about the Ohio outbreak, and I think you should read more about measles here, because I'll probably ask a really nice question about measles. And I just did a quick Google. I wrote measles outbreak 2014. We got Ohio, Texas, New York, Hawaii, Orange County, King County, Washington, and always California. Get your shot. Okay. Influenza. Influenza is so interesting if you want to study viruses. It's zoonotic, and pigs, birds, and humans are a giant mixing vessel of these viruses. They travel back and forth and around, mixing up their genes and DNA, becoming new strains that emerge every year. Why do you think we need a new vaccine every year? Because all the DNA gets mixed up across species. Check out the learning outcomes right here. What happened in 1918? The Spanish flu. What percent of the Earth's population was exposed to flu at that point? How many died? Could it happen again? I want you to tell me, could this happen again? Has it almost happened again? Flu specifically is the influenza. When people say I have a stomach flu, they don't have the flu. The flu, influenza is respiratory. There are two types, A and B. It has a capsule or an envelope that has spikes on it, and these spikes have names, HA and NA, and it stands for hemagglutinin and neuraminidase. Now, I'm giving you H and N because when a new vaccine comes out, it says it covers for H1N1, H3N5, but also in your book you'll see it abbreviated as HA for hemagglutinin and HN or NA for neuraminidase. And see the cute little microbe flashcard in your book. Okay. So why does influenza cycle through populations with varying intensity? It cycles. About every 10 years, there's a terrible pandemic. Well, there's two reasons. One is antigenic drift and antigenic shift. A drift is why we have a different vaccine every year. It's usually trivalent. This year it's quadrivalent because there's 4 different strains in it that they found circulating in Southeast Asia. Drift is very small mutations in the spikes within a single strain. No big deal, minor changes. A shift is major mutations that cause us to make a new number. So if we have antigenic drift, H1N1 is still H1N1. But when you have a shift, it goes from H1N1 to H3N5, or H5N1, which is the bird flu. These huge periodic shifts occur about every 10 years. Your book has a nice diagram showing that. It was called the swine flu in 2003 because the virus had mixed in pigs. It was originally in birds, and then it was in humans, and because birds and humans are both around pigs, they serve as mixing vessels for these viruses. Okay, so take a look in your book. Look at the pages that are here, look at the questions I've asked, and put together a nice explanation of influenza from start to finish. All right, here's a beautiful picture of an influenza virus. Look at these spikes. Neuraminidase, let's start with it. Let's start with hemagglutinin. So hemagglutinin is a protein. It ends with IN. This is what facilitates attachment to your lungs. Once the virus gets in, starts making copies of all the segments of DNA that it carries, RNA. What happens then is when it wants to get out, it has these neuraminidase, which is an enzyme, spikes. And that's what pushes itself out of an infected cell, taking a little bit of membrane with it, and it's ready to infect other people when you sneeze on the bus. This is what it looks like under an electron microscope. And this is a great link from National Geographic. It's only about 4 minutes long, And I would definitely ask questions about that on the exam as well. So got a couple things to think about here. All right, we're nearing the end. Hang on. Rotaviruses versus noroviruses. You ready? Rotaviruses are the #1 cause of infantile gastroenteritis in the U.S. 600,000 deaths in the developing world. Sometimes it's enveloped, sometimes it isn't. It has spikes, and they enter the cell through endocytosis. Almost all children are infected in the first few years of life. It's a fecal-oral route, and the only way to prevent it is proper hygiene, proper sewage treatment, and vaccination. There is a vaccination. Flip the coin over, and you've got noroviruses that infect anyone. Not usually fatal, not usually fatal at all. It's acute diarrhea and vomiting. Cruise ships, they call it the cruise ship virus. Daycare centers, hospital, anywhere people are together and not terribly hygienic. And surfaces, fomites. Fomites are objects that can carry viruses. This is a naked virus. If you remember from the genetics chapter, naked viruses survive longer in the environment than enveloped viruses. Fecal oral route, same as rotaviruses. The incubation is only 24 hours, and it can resolve within 60 hours. Proper hygiene, disinfect surfaces, no vaccination. So take a look at this. Rotavirus versus norovirus. Compare, contrast. We've got 2 diseases that affect and kill infants. What was the first one? RSV. Good. And rotavirus. Okay. I like noroviruses. They're just so rampant and make people so miserable. So I found this great picture on cruiseship.org. So anyway, here are the cruise ships that failed this year so far. And if you like to go on cruises, this is what they found on these cruise ships. Let me just read it for you. Food debris, dead insect droppings, records indicating crew members, including cooks, working while sick. Oh, baby. Gastroenteritis symptoms. That's it. Just go to the bathroom, come back, and keep working. Additional violations included cracked and discarded equipment, which is where microbes hide, soiled cutting boards, food served undercooked. Oh, sign me up. Okay, so you can find all of this out on cruiseship.org. You can find out who's the worst. This guy I like because he says, why is it on cruise ships? Well, cruise ships have lots of people in close quarters. They are floating cities, and because they are required to report every gastrointestinal illness incident. So it's not that it's not happening on land. It's happening on land. It's just not reportable. It can infect as many as 3,000 people a day. It's highly contagious, survives on surfaces, just really nasty. So it can happen anywhere, but it's just more obnoxious when it's on your vacation. Okay, hemorrhagic fevers. Ooh, these used to be really interesting, and I find hemorrhagic fevers fascinating because of the damage they cause. They are just so well suited to really cause horrific disease in human beings. And it used to be that Ebola would just burn itself out because it would strike a village, everybody would sort of die, a couple people lived, it's got like 80% mortality rate, and it wouldn't go any further. No one could walk far enough once they were sick to spread it. And of course now what's happening? Happened in a populated center, people got on airplanes, this virus is adapting. Okay, here's a nice map. I don't know how recent this is. It's pretty recent, so within at least November. When I first saw the Ebola map, it was just up here, and now it's spreading, and we've got more cases. Sierra Leone is now a disaster area. It's just terrible. And look at how many laboratories they have, because hemorrhagic fevers could look like dengue fever. It could look like malaria. They have to send these blood samples away when a person first starts feeling, some malaise and some muscle pain and some fever, and there's not very many places to send it. And then they've got treatment centers, not a lot of treatment centers. It's a big problem. So this is a kinder care center. This is the site of the Ebola restin virus that happened in the book The Hot Zone that some of you are reading. It's now a daycare center. I just find that hilarious. Of course, they've cleaned it out since then. So we all know about Ebola, but what are the symptoms of hemorrhagic fevers in general? They're all fevers, whether it's yellow fever, Ebola, Marburg, or dengue. I did not go into yellow fever. I don't expect you to go into it in your book. There are several types of Ebola. They're all named after where they first occurred. So you've got dengue restin, you've got, Ebola restin, you've got Ebola Uganda, Ebola Zaire, which is the one that's happening now throughout Western Africa. What other diseases present the same as Ebola early on? I already said that's malaria, dengue fever, yellow fever. How are they preventable? This is just knowing what's happening, staying away from where bat droppings are, getting bit by bats. What is the outlook for the current epidemic? You tell me. I just want your opinion. What are people thinking now? The CDC changes its mind every few days. All right, I showed you this before. It hasn't changed. Got any questions? Send me an email. Send me a text. You're all getting better at this. I can't wait for the final. Bye.